Event localization circuitry. You'll recall the PM tubes closest to the gamma interaction within the crystal see the most light and hence produce the strongest electronic signal, while the tubes farther away from the event see less light and therefore produce a weaker, lower amplitude signal. By summing all the signals from all the PM tubes, we get one signal with an amplitude or strength proportional to the amount of energy deposited in the crystal. The amount of energy deposited in the crystal is proportional to the energy of the incident photon or gamma ray. This summed signal is called the Z-pulse or the energy pulse. Additionally, by sending all the signals of the PM tubes through a series of resistors, it is possible to produce two separate pulses which contain information about the location of the photon interaction. These pulses are called the X-Y pulses. The amplitude of the X pulse indicates the location in the X-axis where the photon struck the crystal, and the amplitude of the Y pulse indicates the location in the Y-axis where the photon struck the crystal. The circuit responsible for generating the X and Y positional signals is called a resistor summing matrix. This circuit makes it possible to determine very accurately where the scintillation occurred. In fact, the resistor summing matrix can determine where on the face of an individual PM tube the event occurred. The X and Y positional pulses are used to position an electron beam in an imaging oscilloscope, which then produces a dot on the recording film corresponding to where the photon struck the crystal. Since the collimator eliminates non-perpendicular photons, the location where the photon struck the crystal is directly related to the location of the molecule of the radiopharmaceutical within the patient's body. Correction circuitry. To create an image, we must first correct the electronic signals for distortions which are inherent in any gamma camera detector. To understand how correction takes place, let's start with the X and Y positional signals. As we've just learned, by analyzing all the signals from all the tubes, the resistor summing matrix can calculate where in the crystal the photon interacted. Unfortunately, the PM tube sees light better at the center of the tube than it does at the edges. This has a tendency to distort the signal information such that if a series of straight lines of radiation were placed at the edges of an individual tube, they would appear to be bent toward the center of the tube due to this inherent distortion. This is called a pincushion effect. When the multiple tubes are arranged in an array such as in a gamma camera, the distortions from each tube combine to cause what are called nonlinearities, or simply spatial distortions. The degree of nonlinearity can be measured by placing a bar phantom on the camera, acquiring an image into a computer, and calculating how much displacement from a straight line exists for each line in the image. This is called the linearity of a camera and is usually expressed in millimeters of distortion. Since we're not trying to image straight lines in the patient's body, these small nonlinearities will not affect the resolution of the camera system. They will, however, affect the uniformity of the system, because in areas of the detector where events are distorted, the system will see more events in areas where events are displaced toward the center of the tubes than it will in areas where the events are displaced away from or between the tubes. This will cause hot and cold spots in the image. For this reason, we would like some method of correcting these distortions so that all events can be recorded accurately. Several manufacturers have devised methods of making these spatial or nonlinearity corrections. While each method differs slightly in how it is implemented, the principle is the same. Using a computer, an image of a bar phantom is acquired with the bars parallel to the x-axis of the camera, and again with the bars parallel to the y-axis of the camera. Using what it knows should be straight lines as a reference, the computer can calculate where along each bar in each axis the events are being distorted and how far in which direction they are being misplaced. Then, when the camera is used for imaging, as each event strikes the crystal, X and Y positional signals are sent to the spatial correction circuits, where appropriate corrections are applied depending upon where the event occurred in the crystal. This procedure is called nonlinearity correction, or spatial distortion correction. Now that we've corrected the X and Y positional signals, we can turn our attention to the Z, or energy signal. Just as positional information can become distorted in the detection process, so can energy information. The crystal is not perfect and does not always uniformly convert the same amount of energy to the same amount of light. Because of this, it is possible to have slight differences in the amplitude of the Z pulse depending upon where the event struck the crystal. These differences are known as regional variations in energy response and can also cause non-uniformities in the camera image. To correct these non-uniformities, instead of using a bar phantom, we simply use a uniform source of radiation placed in front of the detector. Special circuits look at small areas of the crystal independently and calculate how each area is responding to the incoming energy. Once this energy data is acquired and stored in the camera's special memory, as patient imaging takes place, each Z-pulse that is created as a result of a detected photon is sent to the energy correction circuitry, where it is then corrected. The combination of energy and spatial distortion correction can produce significantly better linearity and uniformity measurements on gamma cameras than can be obtained without this type of correction. Energy analysis circuitry. Now that we've corrected the X and Y positional signals for spatial distortions and corrected the Z energy signal for regional variations in energy response, we are ready to determine if the event which struck the crystal is valid for imaging purposes. Due to the effects of Compton scattering, radiation originating from a molecule of radiopharmaceutical within the patient's body does not always escape the body with all its energy intact. Many of the photons are scattered as they travel through tissue, losing some of their energy and at the same time changing their direction. If we used every event detected to create an image, we would have a significant loss in resolution due to Compton scattering. Fortunately, when the photon's direction is changed due to a Compton interaction, its energy is also reduced. By analyzing the energy of each photon that strikes the crystal and eliminating those that are of a lower energy than we know the original photon should have been, we can minimize the deterioration in image quality which results from Compton scatter. This energy analysis is called energy discrimination, or pulse height analysis, since the height of the Z-pulse is measured to determine how much energy was deposited in the crystal. If we wish to image only 140 keV photons, we adjust or peak the camera such that it is optimized for detecting 140 keV photons. We then set a window to analyze each Z-pulse after energy correction and reject those that do not have a height equal to the height that a 140 keV photon would produce. Let's examine how this process works in a little more detail. We said that we adjust or peak the camera for 140 keV photons. To do this, we must place a source of 140 keV photons in front of the camera so that we can perform this adjustment. Usually, the technologist will place a syringe containing the radionuclide intended for imaging in front of the detector. Since air produces very little Compton scatter, the photons striking the crystal are, for the most part, all of the same energy. So, if we use a syringe containing a small amount of technetium-99m, we should have only 140 keV photons striking the crystal. By creating a graph of the number of events striking the crystal at each keV, we can produce an energy spectrum of the incoming radiation. For technetium-99m, this energy spectrum will consist of a single peak centered around 140 keV on the graph. The technologist will then adjust the camera so that only events that are within plus or minus 5% of 140 keV will be accepted by the electronics of the camera. This is known as peaking the camera. By telling the electronics to only accept plus or minus 10% of a given energy, we are setting a window. In this case, it would be a 20% window, 10% on each side of the peak. Most imaging procedures utilize this 20% window, although the technologist can set whatever percentage window he or she chooses. The wider the window, the more Compton scatter is accepted, which means the more events are accepted per given time period. This increases the sensitivity of the system and shortens the acquisition times. Unfortunately, the more Compton scatter we accept, the poorer the resolution. A smaller or narrower window would improve resolution, but the accompanying decrease in sensitivity would result in longer imaging times and more patient motion, and thereby negate the gain in resolution. Therefore, the window must be set to minimize acquisition times and still allow adequate resolution for diagnostic purposes. A 20% window is generally an acceptable compromise. In effect, then, we have two factors that are directly affecting the resolution and sensitivity of the camera. The collimator allows only those photons that are traveling perpendicular to the detector to interact with the crystal, and the pulse height analyzer then analyzes the events which pass the collimator to determine if they have been scattered or not. In this way, we make sure that only events that came from a molecule of the radiopharmaceutical located directly under the camera are used to produce an image. Triple peaking. Up to this point, we've been discussing technetium-99m as our source of photons. When an atom of technetium-99m decays, it always emits a photon with an energy of 140 keV. This is not the case, however, with certain other radionuclides used for nuclear imaging. One often used radionuclide is gallium-67, which is used for imaging soft tissue tumors, such as lymphomas, and also for imaging abscesses within the body. Instead of emitting photons at just one energy, atoms of gallium-67 can decay and emit photons at one of 4 different energies. The probability that a given energy will be emitted when the atom decays is governed by the physical properties of gallium. 40% of the time, gallium will emit a photon with an energy of 93 keV. 28% of the time, it will emit a photon with an energy of 184 keV. 16% of the time, it will emit a 293 keV photon, and 4% of the time, it will emit a 386 keV photon. If we only set our camera to accept a single energy, for example, the 93 keV photon, we would be missing 48% of the available photons. But if we could set our camera to accept either of 2 photons of different energies, or any of 3 different energy photons, we could significantly improve our sensitivity. There would be a slight loss in resolution, simply because usually the positioning circuits are optimized for 140 keV, but this loss would be minimal. Most gamma camera manufacturers have added the capability to analyze and accept up to 3 different energies specifically for gallium imaging. This is known as triple peaking and is important in reducing the time required for imaging radionuclides that emit more than one energy of photons. At least one manufacturer now offers 4-peak capability. But this appears to be overkill since the 4th peak of gallium is only 4% abundant, and it is at such high energy that the crystal thickness is insufficient to stop all but a small percentage of the 4% emitted from the body. Counting circuitry. Once incoming Z pulses are analyzed by the pulse height analyzer, if they are the correct height, meaning the photons that struck the crystal were within the acceptable energy range, they are converted into a different type of Z pulse known as strobe or unblank pulse. It's still called a Z pulse, but its height or amplitude is now constant, and therefore the pulse contains no information about the energy of the interacting photon. Instead, the presence of the Z pulse now indicates that the photon was within an acceptable energy range. The Z pulse is now sent to another part of the camera electronics known as the scalar timer circuit. This is simply a device that counts the number of valid events that have struck the detector. Every time a Z pulse is sent to the scalar timer, a counter is incremented by 1. The scalar timer is the controlling device for the gamma camera. Since the image is made up of hundreds of thousands of dots, each representing a single photon striking the crystal, the operator must tell the camera how many dots to put on the film to create the image. This number is a function of the desired image resolution, the length of time available for imaging, the type of study being performed, and the number of events emitted from the patient per second. Typically, each image will contain 250,000 to 1 million counts. As a general rule, the more events detected, the better the image quality. The scalar timer can be preset by the technologist to allow the camera to collect a specific number of events or to collect events over a specific preset time, and, as they are collected, place them on film. By setting the scalar timer, the technologist controls when the camera is creating an image, how many events that image will contain, and/or how much time the acquisition will require. Additionally, the scalar timer usually incorporates a rate meter, which displays the number of valid events that are striking the crystal per second. This is known as the count rate and is useful for estimating how long an acquisition will take to complete. Image recording devices. At the same time that the Z pulse is sent to the scalar timer, it is also sent to the image recording device. For every photon that strikes the crystal, an X and Y pulse is created and sent to the image recording device. The associated Z pulse is analyzed, and if the event is acceptable, a counter is incremented. The Z pulse is then used to turn on the image recording device, and a dot is produced on film corresponding to the location where the photon struck the crystal. In this way, a gamma camera can produce an image of the distribution of radiopharmaceutical within the patient's body. A typical image recording device is called a multi-format camera. A multi-format device consists of a cathode ray tube, or CRT, a lens placed in front of the CRT, some mechanism for positioning an unexposed film in front of the lens, and an electronic control circuit for controlling the image size and position on the film. Multi-format cameras are usually capable of putting 42, 9, or 4 images of varying size on a single sheet of 8x10 X-ray film, with the number being set prior to imaging by the technologist. Many gamma cameras, in addition to being sold with a multi-format camera, will also have the capability to record images on Polaroid film. This option is usually quite expensive in that it requires a separate CRT scope and a special type of Polaroid camera. Because Polaroid film is usually more expensive than conventional film, and because conventional film has more grayscale latitude, the use of Polaroid film has significantly diminished in nuclear imaging. Summary. As we've just discussed, the gamma camera is a device used to detect and record the presence of radiation being emitted from an organ within the patient's body. It consists of various component parts which work together to produce the final image. As the photon leaves the patient's body, it passes through a collimator and interacts with the crystal. The crystal absorbs the energy of the photon and converts it into light. The light produced by the crystal is detected by a series of photomultiplier tubes, which convert the light to an electrical signal, whose amplitude is proportionate to the energy of the detected photon. Special circuits combine the signals from each of the PM tubes to determine where in the crystal the photon interaction took place. X and Y positional signals are produced by the camera detector, corrected for inherent distortions by the camera electronics, and are used to position the beam of a CRT for image recording on film. Additionally, a Z-pulse is corrected for regional variations in energy response within the crystal tube assembly, and is then analyzed to eliminate Compton-scattered events which degrade resolution. If the event is in the appropriate range, as predefined by the operator, the Z-pulse is converted to a Z-strobe or unblank pulse. A counter is incremented, and a dot is placed on film corresponding to the location where the photon interacted with the crystal. In this way, an image is created out of thousands of dots representing the distribution of the radionuclide in the patient's body. By examining these images, the nuclear physician can determine how the organ under study is functioning and can detect the presence or absence of disease within the patient. Gamma camera quality control. What is quality control and why do we do it? Now that we've described the basic gamma camera, its components, and its basic function, we'll discuss the importance of ensuring optimum performance on a daily and long-term basis. Various agencies require that certain quality control tests be performed and recorded regularly for accreditation and licensure of nuclear medicine services. As an example, the Joint Commission on Accreditation of Hospitals has published the following. Standard 3. There shall be quality control procedures governing nuclear medicine services that ensure diagnostic reliability and patient safety. For this reason, it's important to know the basic standards for quality control of the gamma camera. The most important parameters are uniformity, resolution or resolving power, defined in two ways, spatial resolution and energy resolution, linearity or spatial distortion, and sensitivity or efficiency. Uniformity, or flood field uniformity as it is sometimes called, is the ability of a gamma camera to accurately produce an image of the data it detects. It is tested by imaging a uniform source placed in front of the detector. The resulting image should show a uniform distribution across the entire field of view. Changes in uniformity across the entire field of view are called integral non-uniformities and are expressed as the percent change between the area with the least amount of data, the coldest area, to the area with the most amount of data, the hottest area. Integral non-uniformity values represent the maximum deviation from the mean over the entire field of view. Regional changes within the field of view are called differential non-uniformities and are expressed as the percent of change over any given region. Differential non-uniformity values indicate the change in density one sees from point to point within the field of view. Of the two measurements, the differential number is the more important. Integral uniformity in present-day cameras should be less than plus or minus 10% variance, and differential should be less than plus or minus 6% variance. The smaller the percentage of variance, the better the uniformity of a system. Resolution, or resolving power, is defined two different ways when discussing gamma cameras: spatial resolution and energy resolution. Generally, when the term resolution is used alone, it refers to spatial resolution. Spatial resolution refers to the ability of the camera to image two separate objects as they are moved closer and closer to each other in space. If the two objects must be 2 millimeters apart to be seen as two distinct objects, as opposed to one single blurred object, the spatial resolution is said to be 2 millimeters. Spatial resolution is measured by using a device called a bar phantom, which is simply a series of lead bars of a given width and thickness separated by spaces the same width as the bars. Most bar phantoms are known as four-quadrant bar phantoms. They're constructed such that each quadrant of the phantom has different width bars and spaces. Typical bar phantoms will have bars 2 millimeters wide with 2 millimeter spacing, bars 2.5 millimeters wide with 2.5 millimeter spacing, bars 3 millimeters wide with 3 millimeter spacing, and bars 3.5 millimeters wide with 3.5 millimeter spacing. When looking at an image of a bar phantom, it is important to know the bar spacing, because not all bar phantoms are alike. To determine the spatial resolution, the bar phantom is placed on the detector surface, and a uniform source of radiation is placed in front of the bar phantom. An image is acquired and evaluated to determine the smallest bars which are visible. If the image is taken without a collimator, it is said to be an intrinsic bar phantom image, and if it is taken with a collimator in place, it is said to be an extrinsic bar phantom image. Typical gamma cameras will resolve 2.5 millimeter bars intrinsically, or without the collimator, and 3 millimeter bars with the collimator. Extrinsic or collimated resolution is primarily a function of the collimator in use. Another measure of spatial resolution is known as the full-width half-max resolution, and is probably the correct method to accurately determine the resolving power of the camera. But it is a difficult test to perform, requiring a computer and special software. For the purposes of this discussion, you should remember that most resolution specifications are quoted as full-width half-max values, and the typical specification is 4 millimeters full-width half-max. The bar phantom resolution can be calculated by dividing the full-width half-max by 1.75. This means a camera with a full-width half-max of 4.0 could resolve bars that are approximately 2.3 millimeters wide with 2.3 millimeter spacing. Energy resolution is another important measurement of a camera's performance. It describes the ability of the detector to accurately determine the energy of the incoming radiation. Since no camera system is perfect, no system is capable of determining precisely what energy photon struck the crystal. Instead, the system can only determine within a range of values what energy radiation it is detecting. The energy resolution is expressed as a percent of the energy of the incoming photons. If the energy resolution of a camera is 10% and only 140 keV photons are striking the crystal, the system will see photons ranging from 133 keV to 147 keV. That is, it can only determine to within 14 keV what the actual incoming energy really is. Energy resolution is a very important parameter in determining the overall performance of a gamma camera, because it's the parameter which allows a camera to differentiate between primary photons and Compton scattered photons. This ultimately determines the spatial resolution of the system. Energy resolution is expressed as a percent full-width half-max of a specific energy. Most cameras have an energy resolution in the range of 11 to 13% full-width half-max at 140 keV. The lower the number, or the smaller the percent, the better the energy resolution. Linearity, or spatial distortion, is the ability of a gamma camera to reproduce straight-line sources of radioactivity as straight lines. This is evaluated by placing a bar phantom between the radioactive source and the detector. The straightness of the bars is examined. Using special complex programs, the deviation of the bars from a straight line can be determined. This deviation is expressed in millimeters, and, like uniformity, it is measured for both integral nonlinearities, the maximum deviation, and differential nonlinearities, or point-to-point deviations. Three things to look for are: 1. A severe hot rim at the edge of the field of view, or edge packing. 2. Waviness of the bar pattern, which indicates poor differential linearity. 3. Convexities, or bowing, of the bar pattern, which indicate poor integral linearity. These nonlinearities will not significantly impact routine imaging, but become more important when we discuss emission-computed tomography. Sensitivity is a measure of how many photons are detected per unit of radioactivity in a given source. Sensitivity is measured by placing a known quantity of a radionuclide in front of the collimator and obtaining the total counts per minute detected per unit of activity. Sensitivity will change with collimators. In summary, the key parameters to check camera performance are: uniformity, a daily check for uniform field of view; spatial resolution, the ability to distinguish separate objects placed very close together. The two types of spatial resolution are intrinsic, which measures detector and camera electronics, and extrinsic, which measures camera and collimator as a total system. Energy resolution, the ability to accurately determine the energy of the incoming photon. Linearity, the ability to reproduce bars without distortion. And sensitivity, the ability to accurately detect the specified amount of incoming events, or photons, per unit of radioactivity. Computers in nuclear medicine. With the computer, nuclear medicine has taken on a new perspective. Computers can enhance any of the studies we have previously discussed. They can provide additional analysis of the data using mathematical techniques which are more sensitive than the human eye. With the advent of relatively inexpensive, high-speed computers and high-quality image display systems, nuclear medicine has been able to perform new types of examinations that cannot be performed with the gamma camera alone. Examples of these new types of exams are nuclear cardiology and emission-computed tomography, or ECT. A nuclear medicine computer system is a device which provides acquisition, processing or mathematical manipulation, storage, and display of data from a gamma camera. It allows the user to acquire each event as it is detected by the gamma camera and at a later time mathematically analyze these events in the form of images or numerical values. The camera provides an analog signal or electrical wave form representing the event. In the computer, this analog signal is digitized, that is, converted to a numerical value by a device known as an analog to digital converter or an ADC. In other words, the voltage is converted to a number, which represents the location in XY space of the detected event. This number is placed in the memory of the computer. The memory is organized as a square matrix, representing the detection area or field of view of the gamma camera. In this way, each valid photon that strikes the crystal is converted into a number and then stored in a location in computer memory corresponding to the position where it interacted in the crystal of the gamma camera. Images are built up in the computer memory. Events that strike the same location in the crystal are deposited in the same location in the memory. If multiple events strike the same location, they're added to each other in the computer memory location. After acquisition, the number in each location of the memory matrix defines how many events struck the crystal at that particular location. To display the image, the computer converts these individual numbers into appropriate shades of gray or color and adjusts the brightness of the electron beam on the cathode ray tube to represent the individual values. Each location in the image is called a picture element or a pixel. Locations where no events interacted with the crystal are assigned a grayscale value of 0 or black, and the location where the largest number of events struck the crystal is assigned a grayscale value of 255 or white. Depending on what type of film is used to record the image, the scale can be reversed, making the 0 value represent white and the 255 value represent black. Numerical values between 0 and the maximum stored value are assigned values between 0 and 255. In this way, an image is created that has 256 different shades of gray, ranging from black to white. To create a color image, the values in the memory are assigned to a specific color rather than a shade of gray. In this way, individual colors represent areas of the image where the same number of events struck the crystal. The advantage of placing the events into a computer memory is that they can be subsequently analyzed by the numerical capability of the computer. If several images are collected in the computer over time, they can be replayed in rapid sequential fashion to produce a movie or cine display of how the radioactive chemical was used by the organ being imaged. For example, in the segment that you're watching, you can see the radiopharmaceutical traveling through the kidneys. Additionally, because the data has been converted to numbers, we might ask the computer to tell us how much of the radionuclide was detected passing through the left kidney as opposed to the right kidney. For example, if we wish to see how much of the radionuclide was metabolized by the kidneys in the first 3 minutes, we would simply ask the computer to add up or sum the images taken during that time period. As you can see, the computer allows us to manipulate the data after it has been acquired and to obtain more information from the study than if we had simply recorded images on film. Once the data is acquired and analyzed, it can be stored in a variety of ways. The simplest form of storage is to record the image on film. Since the nuclear medicine computer system has a display associated with it that can provide images, if we attach a photographic camera to this display, or send the data to a multi-format camera, we can take a picture of the image. This allows us to save the data on the display, but once saved, we cannot manipulate it any further. For this reason, computers usually have external storage devices associated with them that allow storage of the data in a form that permits it to be loaded back into the computer memory, as if it had just been acquired from the camera. Data stored in this manner can be recalled at a later time and further manipulated if necessary. In the following paragraphs, we'll discuss the various storage devices available for nuclear medicine computers. The most common storage device used on the nuclear medicine computer system is a magnetic disk, usually referred to as a hard disk. It consists of an aluminum platter, similar to a phonograph record, coated with iron oxide. This platter, or disk, is placed on a spindle and made to revolve at hundreds of revolutions per second. A read-write head, similar to the recording head in a tape recorder, is positioned over the spinning disk. As the disk spins under the read-write head, data is either written to or read from the disk. Another very common method of storing computer data is on a floppy disk. This is very similar to the hard disk, except that the platter is made of a flexible mylar plastic rather than of aluminum. Consequently, because of its flexibility, it is referred to as floppy. This plastic disk is coated with the same iron oxide particles as the hard disk and is attached to a spindle like the hard disk. There are some significant differences between the hard disk and floppy that are worth noting. The floppy spins much slower, several hundred times per minute as opposed to several hundred times per second. Even though the floppy disk is read and written very much like the hard disk with a read-write head, the hard disk read-write head never touches the surface of the platter. Consequently, it is very fast since it literally flies above the surface on a cushion of air. The floppy disk, because its read-write head rides on the surface of the disk itself, is slow. Floppy disks are much less expensive than hard disks and can be stored easily. However, they store less information and wear out faster due to the friction of the read-write head against the disk. Another type of computer storage medium is magnetic tape. Often called 9-track tape, this is very similar to regular musical recording tape, only it is about twice as wide. Mag tape, as it's referred to, is the least expensive method of storing computer data, but is very slow in reading and writing. Additionally, it is on a reel and it must be read sequentially. That is, if you want to access data located in the middle of the reel, you must fast forward to the middle before you can read. This is called sequential access and is a very slow procedure. In contrast, disks have what we call random access. The computer can rapidly move the read-write head to any part of the disk and begin to read or write data. This is similar to you moving the needle of a record player directly to the track on the phonograph record to which you wish to listen. One of the questions that often comes up when discussing the various types of computer storage devices is, how many images can I store on a disk? To answer this question, we must first understand the various matrix sizes available in the computer. You'll remember that the image is built up in the computer memory by arranging the many memory locations into a square matrix. And then, as each event strikes the camera, it is placed in the corresponding memory, or matrix location in the computer. This matrix can be thought of as placing a grid over the camera's field of view and placing events in the boxes of the grid depending on where they strike the crystal. The question is, how fine or coarse a grid do we use? The size of the matrix is variable within the computer. For some studies, we use a grid that has 4,096 separate boxes in it. Since we said it was a square grid, this means it must have 64 boxes on a side. 64 times 64 equals 4096. This type of matrix is called a 64 by 64 matrix and is written 64 times 64. If the field of view of the camera is 400 millimeters, then each box in the grid, or matrix, would be approximately 6.25 millimeters square. This means that all events that strike the crystal in that 6.25 millimeter by 6.25 millimeter box will be put in the same location in the computer. In effect, then, we cannot see objects smaller than 6.25 millimeters. So, if multiple events took place side by side in the same matrix location, they would be interpreted as a single event. Consequently, the matrix size that we use determines the ultimate resolution that we are able to see on the computer display. If we wish to get better resolution, we must use a matrix with more boxes, each box smaller in size. If we cut the size of the boxes in our 64 by 64 matrix so they are approximately 3 millimeters square, we have a matrix of 16,384 boxes, or 128 boxes horizontally and 128 boxes vertically. This would be a 128 by 128 matrix. Reducing the size of the boxes by a factor of 2 again, we end up with a matrix of 256, 1.5 millimeter square boxes on a side, for a total of 65,536 individual locations. This is called a 256 by 256 matrix. But what does this have to do with how many images we can store on a disk? Well, we said that each box in the matrix represents one location, called a byte, in the computer memory. So to store an image on disk, we have to store each of the individual numbers that exist in each location in the computer memory. If a computer has 16,384 memory locations, each location being one byte or number, it is said to have 16K bytes of memory. The K stands for thousands. If it has 32,768 memory locations, it is said to have 32K bytes. The memory sizes are usually given in multiples of 1,000 bytes, and normally, computer memory is doubled during the manufacturing process. Doubling the 32K bytes gives us 64K. Doubling this again would give us 128K bytes. Just as memory is referred to in bytes or kilobytes, so is storage space. A typical hard disk might have the capability to store 25 million bytes of information. This would be a 25 megabyte disk. A typical floppy disk can store about 1,250,000 bytes of data and is called a 1.25 megabyte floppy. Tape is generally referred to by how many bytes it can store per inch of tape, since different size reels can hold different lengths of tape. The standard tapes are either low density, meaning 800 bytes per inch, or high density, meaning 1,600 bytes per inch. If an image is acquired into a 256 by 256 matrix in the computer, the image would require 65K bytes of space on the disk for storage. Therefore, a 25 megabyte disk could store approximately 380 separate images. But if the images were acquired in a 128 by 128 matrix, each image occupies 16K bytes. So, a 25 megabyte disk could store 1,525 of these images, or about 4 times as many. Consequently, images acquired in a 64 by 64 matrix would each require 4,096 bytes of storage space, and the 25 megabyte disk would have room for about 6,100 images. Therefore, to conserve disk space, the technologist or physician will decide which matrix size to use for acquisition based on the type of study being performed and the resolution requirements. Well, that was a long way around the answer to the question, how many images can I store on a disk? The answer is, it depends on the type of study you're doing and the matrix size you use for acquisition. In this module, we've discussed the various aspects of a nuclear computer system. We've looked at the analog to digital converters, which take the electronic signal from the gamma camera and convert it into a number for subsequent computer processing and storage. We've looked at bytes of computer memory. We've discussed the computer display and how the data is converted into the various shades of gray, and we've looked at the types of storage: hard disk, floppy disk, and mag tape, used by the computer. All of these parts are known as the computer hardware, the actual physical components that make up the machine. There is, however, another very important aspect to computer systems, and without it, the computer will not do a thing. That important aspect is software. Without the software, the computer is nothing but a mass of silicon, wires, disks, tapes, circuit boards, and so on. It just sits there and does nothing. Software is what makes the computer come to life. It allows the computer to make decisions, to perform mathematics, to display images, to do all the things we expect it to do. In reality, the software is the most important part of the entire computer system. But what is this thing called software? It can best be described as the instructions that tell the computer what to do. In the nuclear medicine computer, software is responsible for telling the computer how to convert each signal from the camera into a number, telling the computer where in its memory to put each event from the camera, telling the computer what value to assign to each location in memory for display of the image in the various shades of gray, and telling the computer when and how to store the images on disk. All of the things that a computer does, it does because it is instructed what to do and how to do it by the software. The software consists of a set of very specific instructions which the computer can interpret and execute. This set of instructions is called a computer program. People who develop the software for computers are called computer programmers and write programs in special languages that the computer can understand. One such language is called BASIC, which stands for Beginner's All-Purpose Symbolic Instruction Code. Because it's a simple language, it's easy to learn, but suffers from limited capability. There are literally hundreds of different computer languages available. But in nuclear medicine computers, the most common language used is called Fortran, which stands for Formula Translator. Fortran was developed for scientific purposes and is very good for performing complex calculations. Many times, when considering the purchase of a computer, an extreme amount of attention is given to the hardware. That is, how much memory does the machine have? How fast can it do calculations? How many buttons does it have? And so on. And the software is completely ignored. This is a serious mistake, for as we have just explained, without software to make it work, hardware will just sit and collect dust. With good software, relatively unsophisticated hardware can be made to perform extremely complex tasks. Unless the buyer intends to write his own software, which is very time-consuming, a purchase decision should be based on the capabilities of the machine, which is predominantly defined by its software, and not strictly on how much hardware a manufacturer can cram into a cabinet.